Dear viewers, greetings. In this video, we shall see about the microbial production of amylase. Amylase is an enzyme that hydrolyzes the starch. It is a glucose polymer that is produced by the plant polysaccharide. It is used in the production of sweetener in food industry. The most important enzyme in the starch saccharification are alpha amylases, beta amylases, glucoamylases, glucoisomerases, fulanases and isoamylases. This is the structure of amylase. Types of amylase: alpha amylases, beta amylases and glucoamylases. Alpha amylase, it is called as calcium metalloenzyme. It hydrolyzes the alpha 1,4 glycosidic linkage in the starch. It faster than beta amylases. Beta amylases, it hydrolyzes the alpha 1,4 glycosidic bond. It is present in inactive form prior to germination. Animal tissue do not contain beta amylase. Glucoamylase, also known as glucan 1,4 glycosidase exo 1,4 glucosidase. It is most effective in acidic environment. Alpha amylase or extracellular enzyme hydrolyzes the alpha 1,4 glycosidic bond. Endoenzyme splitting the substrate in the interior of the molecule. The bacteria involved in the amylase production are Bacillus subtilis, Bacillus cereus, Bacillus amyloliquefaciens, Bacillus coagulans, Bacillus polymixa, Bacillus acidocalarians, Bacillus licheniformis, Lactobacillus micrococcus, Pseudomonas, Artrobacter, Escherichia, Proteus, Thermomonospora, Serratia. The fungi involved in the alpha amylase production are Aspergillus, Penicillium, Cephalosporium, Neurospora and Rhizopus. Among these, Bacillus amyloliquefaciens, Bacillus licheniformis and Aspergillus are highly involved and it produces alpha amylase in a very large quantity and more effectively. Beta amylases, usually it is obtained from the plant origin and even microorganism can produce it. It catalyzes the second alpha 1,4 glycosidic bonds from non-reducing ends. The microbes involved in beta amylase production are Bacillus polymixa, Bacillus cereus, Bacillus megatherium, Streptomyces species, Pseudomonas species and Rhizopus species. Glucoamylases, it splits the glucose from non-reducing end. The microorganisms involved in glucoamylase are Aspergillus niger, Aspergillus oryzae and Rhizopus species. Production of amylase. Fungal alpha amylase. For the production of fungal alpha amylase, the substrate used is wheat bran. The challenges involved in fungal amylase production is the high viscosity caused by the fungal mycelia makes aeration and agitation difficult. Glucose in the medium inhibits the amylase biosynthesis. Inoculum. The spore used for the amylase are Aspergillus oryzae. The media used for the cultivation of Aspergillus oryzae is sabrodextrose agar or potato dextrose agar. The temperature required is 28 degrees Celsius and the duration of inoculum is 5 to 6 days. Medium, cornstarch, corn steep liquor, potassium chloride, disodium hydrogen phosphate, calcium chloride and magnesium chloride. The fermentation condition, the temperature required is 28 to 30 degrees Celsius and the time for the duration for the production of fungal alpha amylase is 3 to 4 days and the pH should be maintained between 3 to 4. Bacterial alpha amylase. The fermentation involved is solid or submerged fermentation, either batch or fed batch culture. The inoculum size is 0.5 ml in 100 ml of sterilized fermentation medium. The inoculum for the production of bacterial alpha amylase is Bacillus species, particularly Bacillus licheniformis, Bacillus amyloliquefaciens, and Bacillus subtilis. Initial media is prepared in nutrient agar land and the duration is 24 hours. Transfer some colonies with sterile needle into the flask containing the sterile nutrient broth. Incubation temperature is 37 degrees Celsius and for 24 hours. Medium soybean meal, amber, distillers dried soluble, N-amine, lactose, magnesium sulfate and antifoam. Fermentation condition. The temperature required is 27 to 30 degrees Celsius and the duration is 48 hours. The pH should be maintained at 6.8. The extraction of amylase includes centrifugation at 3000 rpm for 30 minutes, 5000 rpm for 20 minutes at 4 degrees Celsius. Collect the supernatant for crude enzyme. The condition is 30 to 40 degrees Celsius of temperature. The pH should be maintained at neutral with the help of calcium carbonate. The production begins when bacterial count reaches to 10 power 6 to 10 power 7 cells per ml, typically after 10 to 20 hours, and continues for 100 to 150 hours. Preservation and stabilization. Liquid preparation. Preserve with 5 gram sodium chloride containing less than 2% active enzyme protein. And for solid preparation, it contains up to 5% active enzyme protein. Application. It is used in textile industry, detergent industry, baking industry, sugar industry, treatment of starch processing, waste water, paper industry, biofuel industry and brewing industry. In starch industry, liquefaction of starch for the production of glucose, fructose, maltose. In milling, modification of amylase deficient flour, alcohol, liquefaction of starch before the addition of malt or saccharification. Bakery, increase the production of fermentable carbohydrate. Brewing, preparation of barley, liquefaction of additives, enhanced fermentability of grains, modification of beet characteristics. In paper industry, liquefaction of starch without sugar production for sizing of paper. Textile industry, continuous desizing at high temperature. Feed industry, improvement of utilization of enzymatically barley in poultry and calf raising. Sugar industry, improvement of filter ability of canned sugar juice through the breakdown of starch. Laundry and detergent, increase the cleaning powder additive in dishwasher detergent. Thank you.